effects of mutations. Okay, so we're going to start off with kind of, well, this is going to be a sort of flow diagram. I'm going to start off with a mutation, funnily enough. And let's just remind ourselves that is a change to the base sequence of DNA. Okay, so this mutation can have a few different results. First of all, the simplest result could be that there's no change to the amino acids. And we're going to look at this one. There could be a change to a single amino acids. And there could be a change to many amino acids. If you're not clear on transcription and translation and how the sequence of nucleotides on DNA is transcribed and translated into the sequence of amino acids, then that's your first starting point. I'd go back and check that first of all. So these are the three options. How can we bring about no change to the amino acid sequence? Well, amino acids are coded for by more than one triplet. There's more triplet combinations than there are amino acids. The word for this, which AQA students should definitely be aware of, is that DNA is degenerate. This is mostly going to be brought around by a substitution, possibly an inversion, but again, only AQA students are going to need to know what inversions are. Pretty straightforward. This is hammered into you quite a lot during the course. How do we bring about a change to a single amino acid? Well, this is going to change. The way to do this is to look at, first of all, the DNA. So it's going to change one DNA triplet. As a result, it will change the sequence, or it will change the coding for one amino acid. We're going to follow this through as a flow diagram. So we change the one amino acid. This is going to change the primary structure of the protein or the polypeptide. I'm just going to put here 1'. This is a shorthand for primary. I'm going to write protein just because I'm a bit tight on space. You could say polypeptide. They're kind of interchangeable at this point. So we change the primary structure of the protein or the polypeptide. What happens? Well, we're going to change the internal bonding. The hydrogen or ionic bonding is safest to say. Within the molecule. And this is going to change the tertiary structure. And again, we can write 3' for tertiary in our shorthand. Again, this is most likely to be a substitution doing this. Last column, change to many amino acids. Well, this is going to be the result of a frame shift. And a frame shift is when all the following DNA triplets are affected. So if I draw a little example here and test my memory of triplets and coding for amino acids. Okay, so we're going to do AGT, and that's serine. We're going to do GCC, that's alanine, and CAG is glutamine. This is my DNA, and this is the amino acids they code for. Okay, so if we have a mutation to this, and let's give myself a bit of space. Okay, so let's say, I'll keep my A and my G. Let's say we have an insertion or an addition. AQA students, you're an addition. Everyone else, you call it an insertion. And we're going to add in here an extra G. And now we can carry on as per usual, but we've now got TGC. And we've got CCA, and we've got an extra G. So instead of being serine being our first amino acid, we have arginine. And then TGG is cysteine, and then we have proline, and so on. So all the following amino acids are going to be in the sequence are going to be changed because of this one here. Any addition or removal or deletion of amino acids that's not divisible by 3 is going to result in a frame shift, and it's going to have a catastrophic impact on the polypeptide. So what can we say about this? Well, it's going to change all the DNA triplets. It'll probably change, I mean not 100%, but it's going to probably change all the amino acids in the following sequence. If all the amino acids are changed, well, that's changed to the primary structure. Which is going to change the hydrogen or ionic bonding. Which in turn will change the tertiary structure. So the protein or polypeptide is going to fold differently. So this is going to result from anything that creates a change in the number of nucleotides that isn't multiple of 3. So if you add or remove 6, then it's not going to have such a major impact as doing 5 or 7, because it's going to change the number or the sequence of all the nucleotides in sequence. So this could be addition, is what AQA called it, and for everyone else you call it an insertion. You may hear the term indel, insertion and deletion. That is obviously we group them together because they have a very similar impact. Can also be caused by duplication. Don't worry, everyone else, it's only AQA who deal with duplication. So these are the impacts. So what they're really looking for in the effects of mutations is what's impact is it having to the protein. And so you need to follow through what changes it do to the DNA, what changes it do to the amino acid sequence, and what impact does that have. It could be neutral, it could be beneficial, or it could be harmful. Another word for harmful is deleterious. And this can give a selective advantage, a selective disadvantage, or obviously neutral has no change on the phenotype.